Hello, my name is David Rogers. I'm a scientist at SXT, and along with the team here, we are creating a lab-based soft X-ray microscope. So what's our company objective? Well, we want to bring soft X-ray tomography into every biological research lab. And we want to reduce its cost from hundreds of millions to millions and reduce the footprint, the physical footprint of the machine from hundreds of meters to meters. Well, what is soft X-ray tomography? Well, it's very similar to that of a CT scan of a body, but it's for a single cell, so it allows us to look inside of a cell and create a 3D representation of that cell. And how we're doing that is we're taking advantage of what's called the water window, which is a section of the light spectrum from 2.3 to 4.4 nanometers. And in this part of the light spectrum, water does not attenuate X-rays as nearly as much as carbon does. And so what this gives us is a natural contrast imaging for imaging inside of a cell. This technique is already used. It's used as synchrotrons. There's about 4 full field transmission soft X-ray microscopes in the world, but they have a large cost associated with building and then maintaining the synchrotron, and there's a limited access to the beamlines. So there's a long waiting list for researchers who want to image soft X-ray. So what we want to do is grant that access to every biological researcher. So how we've done that is we're creating a lab-based cryo soft X-ray microscope. So you can see it on the right is our pilot system, and it has a footprint of around 2 by 2 meters, and it doesn't have any special requirements. So it can fit in any lab that takes an electron microscope. So who's interested in soft X-ray tomography? Well, a lot of people, disease researchers, are already using this technique to look at the morphological changes in a cell when it gets infected with a virus or if it has a neurodegenerative disease. People involved in the pharma industry who are doing drug discovery are using this technique to make sure that drug delivery mechanisms are working correctly, and that can really shorten the path from discovery to drug development, saving a lot of money. And biological researchers really want to use this technique because it has a couple of advantages. It can do whole cell imaging, unlike an SEM or TEM, and it has a higher resolution and contrast than that of visible microscopy. So when used in conjunction with those techniques, it can really add a new layer to the knowledge of a cell. So how does a soft X-ray microscope work? Well, you have a soft X-ray light source, which is then focused down onto a sample. The sample has the ability to rotate so that you can get a 3D projection of it. The X-rays travel through the sample and onto a zone plate. That zone plate then focuses down the light onto a CCD, giving us an image of our sample. So what is our soft X-ray source? This is the heart of the system. So we have a laser produced plasma on a solid target, and the reason that we're able to do that is that we have self-healing illumination and collection optics, and this gives us the ability to get a high numerical aperture and to have a very tight small focus, which gives us a very small plasma source. And this is really necessary for having a good coupling with the microscope optics. Since we're also using metal fuel, it gives us a broadband source, and so we're able to have a choice of wavelengths within the water window. So what are we using for our microscope optics? We're pushing the boundaries on X-ray optics, and we're using ellipsoidal grazing instant optics, and the challenge with them is that we need to keep a very low high spatial frequency roughness and a very low shape error so that we have an efficient transport of photons throughout our microscope. We're using multi-layered optics to monochromatize our broad X-ray light, and using that we can also select different water window wavelengths. And we are using zone plate for our microscope objective to focus down onto the CCD. And we're choosing, we have to choose materials carefully to maintain the highest efficiency that's physically possible. So what's our resolution? We're getting down to below 60 nanometers. On the left is a Siemens star, which is a sample target to measure resolution. And so we are seeing contrast levels around 26% at 60 nanometers. So that resolution is comparable to diamond synchrotron, which is getting down to 30 nanometers on a Siemens star. So here's some images. We have an SEM on the left of diatom. A diatom is a type of algae. On the right, we have our 2D image of a diatom, of three diatoms. And you can see that we're able to image inside of a diatom or through the diatom and see the holy carbon beneath it. These are dry images. We also have cryogenic images of Chlamydionis algae. So on the left is a 2D image, and you can see a structure inside the algae. And on the right is a 3D projection of an algal shell. The blue is ice, and the green is grid, and the red is our algal shell. So what's next for Sirius XT? Well, we have a pilot system created that we're going to be delivering to the Conway Institute of Biomolecular and Biomedical Research in UCD, where we can image samples there. We're building a new system, and we hope to have a 2022 microscope product launch. We also have lots of improvements that we're working on. So we are going to be integrating a fluorescent imaging microscope so that we can image inflorescence and soft X-rays on the same sample. We also are implementing changes that are going to allow us to use different types of samples, so we'll be able to use capillary and TEM grids. We also have new laser optics and stability improvements that are going to improve our imaging time so that we are close to that of a synchrotron. Thank you.